Hello everybody, my name is Vance, and today I want to get a little scientific. So, there's a process that we all do every day, millions of times a day actually, and we never even realize it. It's one of the most critical functions to our daily survival and pretty much everything we will ever do, down to how we look. I'm talking DNA replication. This is actually one of the most critical and important processes your body can do, besides, well, breathing and digesting food and water for nutrients, but that's for another topic. So DNA replication is a process that occurs in every cell whenever it goes through cell division, which I may talk about a little more in depth in another video. Today I want to focus on just this. So, first off that I kind of need to cover, origin of replication. Our DNA is so long and so big, it will have multiple bubbles form that basically will separate all of the hydrogen bonds connecting all of the nucleotides within the DNA. And this is known as our origin of replication. So, now that we've got this separated, we've got a couple things keeping it, breaking apart. I just realized I forgot to. So there's a couple things at play here. A couple of enzymes and a couple of proteins. So the first thing we need to know about are what are known as helicases. That's one. And helicases are little, well, basically they're like zippers. Best described, they're like little triangles that push through and they will break all of these bonds, holding all of the nucleotides together. And for those of you who don't know, the four basic nucleotides are adenosine, thymine, guanine, and cytosine, represented by here. Now, each one of these has a complement to another. So every A will be matched up with a T, every G matched up with a C. An easy way to remember this is apple tree chewing gum. So, these are what's known as helicases. They're enzymes, and fun fact, every enzyme will end in ACE, so that's an easy way to tell them apart. And they will actually break all these hydrogen bonds holding the DNA together in order to split these two apart. Now, that's all fine and dandy, except these DNA strands kind of want to come back together. So we get what's known as single-strand binding proteins that link up here, here, and here, and they kind of just follow the they follow the helicases. What these do is they just keep the DNA from recombining. Simple as that. So single-stranded binding proteins. A little bit of a mouthful. And one more challenge we have to overcome is our DNA, it's twisted. It's a double helix. And we need it to not be a double helix, because as these go through and they break all these hydrogen bonds, that DNA is actually going to twist and knot. It's going to be even harder and probably impossible to break through. So we have one final mechanism set in place to keep our DNA from raveling up like that. And they actually fall up ahead of the helicases, and these are what's known as topoisomerase. It's a bit of a mouthful. Topoisomerase. Now, if that seems a little too much to remember, that's fine. It's also known as DNA gyrase. Why they gave them two names, I don't know, but it's known as topoisomerase, these little enzymes right here and here, and they kind of just move outward. But it's also known as DNA gyrase. So those are the basic enzymes that kind of split it apart. Now we talk about replication. Now the first thing we need to know is this first initial break. Our first initial break was about right here on this diagram, and we actually get what's known as special forks. So before I talk about that, I need to discuss real quick that all DNA strands have a beginning and an end, more or less. So this end is our 3' end, and right here is the 5' end, and over here is the 3' end and 5' end. Now it should be noted that DNA strands are anti-parallel. Basically, they're running in the same direction, they're just going opposite directions. So at this fork, at the origin of replication, we actually get two new ends. So now we have 5'3", 5'3", and 3'5", 3'5". So here's where the replication begins. Like right here, actually, we'll go with. So an enzyme called primase will come in and will fill in the blanks. And now it should be known that this is actually RNA. This is not DNA, so this is not going to stick around forever because it would kind of not be good if we had RNA mixed in with our DNA. Primarily because RNA replaces the T, the thymine, in DNA with Uracil. We can't really use Uracil in our DNA for a lot of reasons that I might talk about in another video, but we'll get to that more later. So anyways, it will fill in these blanks. So T to A, C, G, G, C, C. Yeah. Alright, so this is just RNA, and that's what the job of the primase was. So now, the what's known as, I believe it's DNA polymerase, yeah, that's right, so I had to do a quick double check. We'll go in and we'll actually fill in the blanks. And here is why we can't just do a duplication like a normal cell division. See, it would be almost impossible, actually it is impossible, for us to just do a duplication of the DNA like a photo scan or anything like that. Because how DNA replicates is it'll take the strand it already knows. So right here we just have this strand up top. So the body can look at this and say, oh, there's an A. T is complementary, so we have to put that as a T. So that's why we can't just do a quick photo scan and, duplicate it like that. So basically our polymerase, particularly DNA polymerase 3, will come in and it will fill in the blanks. Now it should be noted that it always fills in this way. So without any interruptions, without any interruptions, and of course the helicase is in my way. I keep saying without any interruptions. It will fit, the DNA polymerase will go in and fill the blanks here. C, A, yada yada yada. Now we do run into a slight problem though when we try to go the other way. Because the DNA will always replicate running 3 to 5. So here we run into a bit of a stalemate. Now here's what we do. Our bodies will say, let me just break those for ease of access later. Our body will go through and they'll come and they'll jump back a couple and they'll write in C. They'll write in a couple of these with using the primase. And then our DNA polymerase will go through and fill it out. So TCA, A, G, T. Now, of course, we still have a long ways left to go, so we've got to do the same thing. We'll jump back, and we'll get, or wait, no, cannot be T, because remember, RNA has the uracil, which is U instead of thymine. So we'll replace that with U, A, G. We'll do the same thing, C, A, T. And it'll do that all the way until it gets to the end. And then we run into a bit of a problem, which I'll get to in just a minute. And then the same process is repeated on the other side. Now, this right here, this is a division. Think of these as two separate strands. Right here, this is what's known as the leading strand. Okay? Because it's able to just keep going and going and going. This, however, having to do it like this is the lagging strand. And since these run anti-parallel, whatever is up here will be the opposite of what's down here. So running this way will be the leading, and this way will be the lagging. Okay? So, once we've done all of that, we hit a bit of a snag. Remember, we can't have RNA in our DNA, right? So, I believe it is DNA polymerase 1. DNA polymerase 1. So that's D. Spelled exactly the same ways. Polymerase 1. We'll go through and essentially replace the RNA. Pretty simple. It'll just replace the RNA throughout the entire strand, and that's pretty much it. However, here we hit a bit of a problem because there's actually a bit of a gap right there, and right there, and right there. These little chunks of information of DNA, this is what's known as an Owazaki fragment, named after the Japanese scientist who discovered it. So, this DNA strand is not complete. So we actually have a protein called ligase go through and will actually basically seal up any gaps that there might be. And that's pretty much it for DNA replication. Once that's all done, it goes through the entire strand. You're ready, it sends it off into the new cell to be, well, divided again sometime, eventually, maybe, I don't know. That's the basics of DNA replication. Now, there is one other problem that we run into. And let me just cut to a quick erase of this, and I'll explain that when I get back. Okay guys, so I'm back with the fresh drawing here. So let's pretend that this is actually the end of the DNA strand. Okay, it cannot go any further. Now, if you're the leading strand, then you really don't have anything to worry about. You're fine, you're going to be good, you know, it'll replicate normally. So, you know, you'll end up with the A, C, T, G, C, A, G, T. This is all fine. However, the problem comes when you're having to go backwards because your DNA wants to replicate 3 to 5. Well, it can't do that. It can't just go in a normal direction. So, here's the problem. So, say that, you know, you've got your U, G, C, and then of course your DNA replicates as normal, you know, in a normal lagging strand. So you have your A, your G, T, A, T, C. Now normally it would be able to back up and you can continue on. Well, there's a problem. We can't back up anymore. There's nowhere to back up. So this chunk right here is pretty much lost. That's fine because the end of DNA strand all falls in line with the chromosome at least, on what's known as a telomere. And when we think chromosome, we normally think, oops, messed that up. When we think chromosome, we normally think something like this, right? Well, telomeres are specifically designed to be that tip, those tips there. Those are the telomeres. They're basically the end of the chromosome, and they don't really have a purpose other than giving us leeway. Because every time this happens, we lose that chunk of DNA. It's unusable because there's no complement. Basically, there has to be a complement for it to be usable, even though it's still kind of there, but most of the time it just falls off and dies. So over time, these telomeres will shorten. And this is a process that scientists believe is directly involved in aging. So the chromosome, which actually by itself looks something like this, although maybe not so thick, just looks something like this. This is, let's say this is the chromosome of a 10-year-old versus the chromosome of a 50-year-old. It's noticeably shorter, just because over time and through all these cell divisions of which we've lost this DNA, those telomeres have gotten shorter. So telomeres are something that we believe are involved in aging. Now, there's one more thing that I need to talk to you about, that I want to talk to you about today. It's still something known as telomerase. Or telomerase? I am probably saying that so wrong. Yeah, I believe it's yes, telomerase. There's no A there. So it's telomerase, and this is a kind of funky but pretty cool enzyme, because of course that A's. So how telomerase works is it basically extends the chromosome. It basically makes it longer. Now, this may seem like a very, very weird process, and possibly impossible process, and you'd probably be right, except a lot of our cells do it on a daily basis, particularly cells that have to replicate a lot throughout the day. Like any cells that's constantly dividing are having to do this process using telomerase. So basically, telomerase has a set pattern. Let me make sure I'm still on camera here. That'd probably be weird. I'm not. So telomerase has a very set pattern. Essentially, it's going to connect a predetermined useless pattern of RNA, I believe, RNA, onto the end, or the 3 end of the DNA strand. So basically, this pattern will be very predictable, basically it's useless, like I said. So it's going to be something like A, T, 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 G, etc. That's etc. Stop saying etc. or however you say it. So it's a very predictable pattern, and it'll basically just connect on to the end of the 3 end. So you know, A, T, 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 G, G. So even though these are useless, this gives the DNA space, or the primase space, to back up and say, okay, well, I'll just attach some pointless stuff there, and then we can finish what's there. So all of this is basically leeway, saying, hey, I mean, we're useless, but, you know, once you divide, this chunk's going to get chopped off, but you'll still have all this, so you're not losing any genes. And that's one of the main problems with cell aging and cell dividing. These telomeres, they can only be shortened so many times before you'll actually start cutting into the genes, which is bad for obvious reasons. But otherwise, there's not a whole lot, really much more. Those are pretty much the basics about DNA replication. I'm sure I missed something. I'm sure I did something wrong. If I did, feel free to call me out on it. We're all here to learn together. If you did enjoy this video, consider giving it a thumbs up. If you have any ideas for anything I should talk about in the future, leave a comment down below and just let me know. And if you know, you have anything else you'd like to say, any comments, any concerns, and like I said, if I did anything wrong, feel free to call me out on it. If you have any further questions on this, also feel free to leave a comment down below, and I will be more than happy to answer them. But thank you all so much for watching. More videos will be headed your way, and I will see you in the next video.